Okay, this video is about the sort of underpinning concepts in biological molecules. We're going to deal with quite a few biological molecules over this coming term. We've got the carbohydrates, we've got the proteins, lipids, and finally we've got the polynucleotides of the nucleic acids. So, a couple of terms that you might see sort of lurking around in books and in booklets are monomer, dimer, polymer and macromolecule. So if you think about a monomer, mono means one, and so if we're talking about a monomer, we're talking about a single unit of a molecule. And these are usually given in the carbohydrates. These are called mono, meaning one, saccharides. And these are the sort of very simple single units of sugar, and a good example of that would be glucose. Other ones you might have heard of are fructose, and we will look at the monosaccharides in more detail. Now, in proteins, when you did proteins at school, you did them in the terms of digestion, and you know that proteins are broken down to amino acids, and so the monomer of a protein is an amino acid. And the monomer of the nucleic acids, like DNA, are nucleotides. So we've got monosaccharides for carbohydrates, amino acids for proteins, nucleotides for the nucleic acids like DNA. Dimer, you will not be surprised to learn, is where di means two. We join two monomers together, and the sort of general term for what you get is a dimer. And again, I think you'll see a bit of a pattern emerging here. For the carbohydrates, these are called disaccharides. And a good example of that might be something like maltose. You actually need to know 3 disaccharides, so again there'll be another video dealing with that. Slightly differently for amino acids, when we join two amino acids together, we call them after the bond that's formed. So we call them di, meaning two amino acids, and the bond is a peptide bond. So these are called dipeptides. And you'll never come across a situation really where two nucleotides are joined together. They're always joined together in huge long chains. I suppose the correct appellation for the dimer of a nucleotide would be a dinucleotide. Don't think you'll come across that. So, if we add more monomers to our dimer, and just make a dimer, and if we join more monomers to that, we'd have many of them. And the prefix meaning many is poly. So here we're talking about polymers, so repeating subunits. And again, in the carbohydrates, these are called polysaccharides. For example, starch, which you'll have come across at school, which is a storage polysaccharide, or cellulose, again you'll come across that in the context of cell walls. It's a structural polysaccharide. In terms of proteins, when you join a lot of amino acids together, you get a polypeptide. When it folds up to form a shape, for its function, it's then called a protein. So just a big chain of amino acids is called a polypeptide. And again, this won't be a big surprise. In the nucleic acids, there are polynucleotides. So I said at the beginning we were going to deal with 4 different types of biological molecule. So we've got the carbohydrates, which are coloured in red for you. We've got the things about proteins, which are in this sort of bluey-green colour, and things about the nucleic acids. And the one that we're missing is lipid or fat. And lipid or fat is a big molecule. Obviously polymers are big molecules because you've got loads of monomers all joined together. But these are big molecules without repeating subunits. So a macromolecule, macro means big. Macro is a big supermarket. And here we're talking lipids or fats. And lipids and fats are called macromolecules and not polymers because, as you'll remember from GCSE, they are formed from glycerol, which is not a small molecule, which is a small molecule, and 3 big molecules called fatty acids. So they all form big molecules which are sort of bunched together to form lipids. So let's just look at the 2 major reactions in biology, and these are to form dimers and polymers and to break them down. So we've got 2 types of reaction: a joining reaction, which is condensation, and hydrolysis, which is a digestion or breaking down reaction. So this one's used to join molecules, and this one is used to break down or digest molecules. So condensation always makes me think of water on glass. So if you hot summer's day next year, you might be sitting in the garden, you get some lemon squash, put it in a glass with some water and put some ice on it, and you'll get condensation from the air forming on the outside of the glass. So a condensation reaction is to do with water. So on one of our monomers, you would have an OH group, and on the other you'll have a hydrogen group. And if we join those 2 together, the idea is that you eliminate H2O. So 2 hydrogens and 1 oxygen out of the bond. So condensation is elimination of water. And of course the molecules that are left that were bonded to the oxygen and hydrogen will then form a bond. And those bonds all have names, and as you go through the videos we'll deal with those. The opposite of that is to take a dimer or a polymer, doesn't matter, anywhere where you've got a bond. Just join my dimer together, and to chemically insert the water. Now it's important that you don't just say adds water. If you add water to a dimer, dimers are quite small, lots of them are quite polar, it'll just dissolve. So this is chemical insertion of water. So what we're doing here is we're taking H2O, we're making it back into an OH group, and a hydrogen group, and we don't care how that's done because we're not chemists. And we're adding back in the OH group to one and the hydrogen to the other, and that bond will break. Okay, I think that's the basics, and see you next time for monosaccharides.